Hello everyone, and thank you for the invitation. I'll be presenting my work on human brain development, a cross-sectional and longitudinal study integrating multiple neuromorphological features. So, studying the stages of brain development, it's been known for a long time that brain undergoes a massive cortical structural remodeling that starts from childhood, which continues well into the third decade of life. But surprisingly, looking at the huge body of the neurodevelopmental literature, most of our understanding of brain development is derived from studies examining brain development in the context of single morphological measures or studying them in statistical isolation, while ignoring the intrinsic interdependencies between these measures, such as cortical thickness, cortical surface area, or cortical curvature metrics. And as a result, we know a lot about these measures and how they develop in the absence of other measures. However, given the dynamic biological relationships between these distinct morphometric properties, and importantly, each carrying specific yet complementary neurobiological information, it is also crucial to study their interrelatedness in the context of typical cortical maturation. So here we propose to use a novel implementation of a multivariate technique, non-negative matrix factorization. And so basically NMF is a matrix decomposition technique that decomposes the input matrix into patterns of covariance or components and their weights. And this technique has been recently explored in the context of neuroimaging, pioneering by Dr. Sotaris and Dr. Saragenan to capture dominant patterns of covariance. And more recently, this method has been adapted and developed in our group to enable the integration of multiple structural metrics. And as in the present work, so the novelty of our work is that we've been adapting the technique to stack up the different morphometric features of subjects, and then the way which we do the decomposition gives us two matrices. So one is a vertex-wise matrix, which gives you a weight at each vertex across a number of user-defined components, and then we can look at how each of these components gets a specific contribution from cortical thickness measure, surface area measure, gyrification index measure, and mean curvature measure across all subjects that we are analyzing in our data. So based on this, the objectives of this work is to first to implement NMF, to investigate the interrelatedness of cortical morphological measures using a cross-sectional data sample, and secondly, to investigate patterns of coordinated change across multiple measures of morphology using a longitudinal data sample, and lastly, to investigate how such patterns of morphometric covariation might be influenced by variation in demographic and cognitive ability. And to do so, we've used a sample of structural T1 MR images of typically developing participants aged 5 to 25, provided from our collaborator at the NIMH. And we used 776 participants per scan for cross-sectional analysis, and a longitudinal subset of this data consisted of participants each with 3 scans was selected for the longitudinal analysis. And notably, this dataset included participants' socio-economic and cognitive data. So moving to the NMF decomposition results, in the cross-sectional analysis, NMF identified 6 different components that we derived from our data, and each component is basically a cluster of vertices that tend to share a specific pattern of covariation across subjects and morphometric features. And here we have the subject per metric weights organized for each metric across the different components, which describes a relative weighting of each subject on each component by measure. So here you see the variation in subject weights, which are organized in an age-ascending manner per metric. And for example, you can see how cortical thickness weights are lower in older subjects, and also you can see the overall subject's variation weights across metrics. So some interference that we can make here is that, for example, component 2 is characterized by higher values of cortical thickness and mean curvature, moderate surface area variation, and lower values of gyrification index in these specific cortical regions. Whereas component 5 is more about higher surface area weights in primary heteromodal regions of the inferior temporal and parietal cortices. Moving on to the longitudinal NMF implementation, here we specifically used age-related slopes across 3 different time points derived from each subject at each vertex for each metric. And this implementation will result into a different interpretation that rather highlights the velocity of change, in which lower weights are interpreted as steeper decline of a metric, while higher weights are interpreted as a relative preservation. So looking at the longitudinal NMF results, here these components have captured vertices that share a same pattern of synchronized change across these measures, which the pattern then can be described from the weight matrix. So for example, again here, we can see that the longitudinal component 2 describes relative preservation of cortical thickness, a sharp decline of gyrification, and a decline of surface area and mean curvature in bilateral, occipital, and temporal poles and the right primary somatosensory areas. So after identifying these components of covariation across both morphometric measures and their coordinated change, so next we asked if these patterns might have significant demographic or cognitive mappings. And to assess that, so here we have the morphometric component pairs on one matrix and the demographic variables in another matrix. And what we chose to do was to decompose these further to find morphometric signatures associated with these inter-individual variations using partial least squares, which basically gives you linked patterns of brain and behavior covariance in latent variables. And here I'm showing the results of the one significant PLS latent variable derived from cross-sectional analysis that highlights maximal correlation across these 2 sets of data. And on the right-hand side, the brain maps are summarized with their specific morphometric profiles, and each significant morphometric feature contributing to this latent variable is displayed with its bootstrap ratio. And on the left-hand side, we have the significant demographic features contributing to this latent variable, which are highlighted in yellow. And so this latent variable accounts for 96% of the covariance, and it is very much an age-related latent variable, showing older ages to be associated with this distributed decreases in gyrification index, cortical thickness, and surface area throughout the cortex, and also local increases in the sulcal depth in mean curvature. And we do also observe the subtle impact of lower socioeconomic status and IQ scores on these patterns. Next, the behavioral PLS analysis of longitudinal NMF revealed 3 significant latent variables accounted for around 97% of the covariance together, from which I'm just going to show you the first. So this latent variable explained 47% of the covariance across the brain and demographic data, which loads dominantly and more preferentially onto females and suggests that more socioeconomically advantaged and high IQ females accelerate through neurodevelopmental processes. And this pattern is mostly manifested in surface area and then cortical thickness almost uniquely in regions that are related to heteromodal processes. On the other hand, mean curvature changes in longitudinal component 6 exclusively shows a more stable or increasing rate of change in these individuals. And notably, comparing the results from the 2 PLS analysis of cross-sectional and longitudinal NMF weights kind of emphasize that the inter-individual variability in factors such as age, sex, environment, and cognitive ability are better explained by variations in tempo of anatomical change than cross-sectional morphometric covariation. So finally, just in conclusion, we identified patterns of cortical covariation across integrated measures of cortical morphometry and their synchronized rate of maturation in typically developing youth. And we observed a non-uniform relationship between morphometric measures and their coordinated maturation throughout the cortex, which underlies fundamental neurodevelopmental processes that co-vary together in this period. And we observed that these patterns are age-related, are sexually differentiated, and have a strong association with individuals' variability in socioeconomic privilege and cognitive ability. And taken together, we believe that this novel characterization of cortical morphometric features maturation would not only contribute to better modeling the typical brain development and its underpinning mechanism, but it also shapes thinking about mechanism underlying pathological neurodevelopmental instances. And with that, I'd like to thank you very much for listening.